Thanks. Thank you, Yang and Andrew, for inviting me to present our poster here. So I have nothing to disclose. My laboratory is really interested in this pathway, the IRE1 XP1 pathway of the endoplasmic reticulum stress response. As you can see, this whole study from this protein called IRE1 that sits on the membrane of the endoplasmic reticulum. And when you have this oncogenesis process going on, this IRE1 would take its enzyme activity that excites 26 nucleotides from the already mature XP1 mRNA. And due to the translation frame shift, now they will this mRNA, the splice mRNA, will encode a bigger 54 kD functional transcription factor that could enter into the nucleus, upregulate and downregulate about 500 genes, and allow them would have to do with supporting the growth and survival of CLL. So my laboratory has been knocking out the XP1 gene from the emu-TC1 mouse model. As you can see here, when you now get the B cell population and analyze for the low B220 and CD5 positive CLL population, you can see XP1 knockout mice, XP1 knockout emu-TC1 mouse, they would actually have slower CLL progression, as you can see here, and the mouse actually also live longer. And my laboratory also developed an inhibitor called BI09 that we use to target the IRE1 RNA activity, and therefore allowing us to control the XBP1 transcription factor. And very interestingly, while we were studying the XBP1 knockout emu-TC1 cell, what we have found out is that when XBP1 is not there, now IRE1 begin to cleave the mRNA of the secretory immunoglobulin heavy chain, because this immunoglobulin mRNA also have a signature very similar to XBP1. So now IRE1 will cut this secretory immunoglobulin mRNA one cut and let it to die, right? So now by looking at this data, it's really quite surprising. Initially, we thought now when you don't have XBP1, IRE1 will go to the roof, and that's due to the fact that now you don't have XBP1, so the cell don't produce secretory immunoglobulin. But turned out that's not the case, because when you knock out IRE1, therefore you also don't have XBP1, but still these B cells produce secretory immunoglobulin, mRNA, and also the protein. And if you now bubble knockout, in XBP1 knockout, you also introduce IRE1 knockout, now this immunoglobulin comes back. So this suggests to you that IRE1 can indeed cleave the mRNA of the secretory immunoglobulin. So what does secretory immunoglobulin have to do with CLL? It turns out that in order to understand this, we decided to cross the MD4 mouse model with emu-TCL1. In this case, now the B cell will produce a lot of antibody IgM against heniglysosome, and you indeed see the mouse actually survive much shorter, have about 8 months median age. And surprisingly enough, if you now look at their blood, bone marrow, or spleen, now these mice, they begin to accumulate tons of myeloid cells, CD11b positive myeloid cells. And these majority of these cells are actually also Ly6G positive, suggesting they are granulocytic myeloid cell. So these myeloid cells, we call them MDSC because they are capable of suppressing T cell proliferation, as I show you here. So to further suggest that indeed secretory IgM can indeed promote MDSC to promote the function of MDSC to encourage CLL progression, we decided to cross the mu-S knockout mouse model with the emu-TC1 mouse model. So in this mouse, now they cannot produce secretory immunoglobulin, and you can see now the mouse live much longer. They have about 14 months median age, and now they have dramatic decrease of the myeloid cell, and also the MDSC from these mice, they cannot suppress T cell proliferation. And finally, I just told you about RIDD. Can we target XBP1, therefore activate RIDD to shut down secretory immunoglobulin to control MDSC. Turns out that we can do that. So now you can take MDE4 emu-TC1 mouse, cross it with XBP1 knockout, and in this case, you do see downregulation of secretory immunoglobulin, also much lower myeloid cell. Interesting enough, when you knock out XBP1 from CLL cell, what you have found is that myeloid cells actually reduce their expression level of arginase-1 and IDO, which are very important for their function in suppressing T cell. So these are the data showing that indeed, myeloid cell from the XBP1 knockout MD4 emu-TCL1 mouse, they are much less capable of suppressing T cell, and our inhibitor can phenocopy genetic XBP1 knockout mouse model showing that when you treat the cell with the inhibitor, IRE1 go to the roof, XBP1 is shut down, and you also have less secretory immunoglobulin. And in this case, if you take myeloid cell from the inhibitor-treated mice, you could also see they are much less capable of suppressing T cell. And finally, I just want to show you this data showing that B cell in general produce IgM to activate MDSC. In this case, we take wild type and MUS knockout, planted these mice with Lewis lung cancer, and you could see Lewis lung cancer grow a lot slower in the MUS knockout mice, and the MDSC isolated from tumor indeed lose their function in suppressing T cell, and we know it's really CD8 T cell because if you use anti-CD8 antibody to deplete the CD8 T cell, the tumor grow back. So I thank you very much for your attention. I would love to talk to you at our poster section and answer your questions.
Thank you very much.